Check.
Are we on? There we go. We're live. Good morning, everybody. Hope you've all had a chance to help yourself to the strawberry kiwi infused water and other refreshments outside. And welcome to the first of our Grand Round series of the new academic year. And it's a great turnout for that. The summer days here. I'm going to be introducing our introducer, as is the tradition here, in just a minute. First, let me also remind you, though, that after the conclusion of the Grand Rounds today, there is a session across the hall in the conference room for trainees. Even if you didn't RSVP, if you're a trainee or were once a trainee or want to be a trainee again, you're all welcome to be there. So with that, let me introduce first Jordan Cohn, a postdoc in Susie Hong's lab, who's going to be doing the formal introduction for our speaker.
Thanks, Neil, and hi, everyone. I'm Jordan Cohn, and I'm a postdoc in Dr. Susie Hong's lab. And I'm pleased to have been invited by Susie and Neil to introduce today's Grand Rounds speaker, Dr. Andy Miller. Andy is currently the William P. Timmie Professor and Vice Chair for Research in the Department of Psychiatry and Behavioral Sciences at Emory University School of Medicine in Atlanta, Georgia. Dr. Miller received his medical degree from the Medical College of Georgia and completed his residency and fellowship in biological psychiatry from the Albert Einstein College of Medicine in New York, where he then went on to direct the psychoneuroimmunology research program. Andy is an internationally recognized expert in brain-immune interactions as they relate to depression, having demonstrated that inflammatory cytokines can disrupt dopamine and glutamate metabolism in the brain and alter neurocircuits relevant to motivation, reward, and anxiety. In addition, Dr. Miller and his group conducted the first clinical trial examining the efficacy of immunotherapy for the treatment of depression. Andy has produced over 250 scholarly publications, won numerous research, teaching, and mentoring awards. He also served on my dissertation committee and is an ISI highly cited researcher and a board-certified psychiatrist. Please join me in welcoming Dr. Andy Miller.
First, push something on. Does it work? Can everybody hear me? Fabulous. Jordan, thank you very much. That was very sweet, very kind. And Jordan was a real joy to work with at Emory. And he is, I hear, a little bit of a diva in the lab. And Susie's having a lot of fun with him. But he was so excited to be able to come to San Diego. I remember I wrote a letter to you and said, Jordan's a great guy, which it's glad he made it here, and thank you, Jordan. I appreciate that. It really is an honor for me to be here and have an opportunity to share with you a little bit of the work that we do in terms of the impact of inflammation on the brain, and especially as it relates to depression. And I think most of you are aware, just to kind of put this into a context, that depression is a devastating disease. It affects about 10% of the adult population in the United States. It's the 10th leading cause of suicide, and just recently the World Health Organization deemed depression is the leading cause of disability worldwide. But for those of us who treat depression, probably the most vexing issue is the fact that there's a significant percentage of people who do not respond to our conventional antidepressant treatment. And that includes not only the pills that I've indicated here, but also psychotherapies as well. And so what that really emphasizes for us is that we really need to start thinking outside the box. We need to look at different pathophysiologic mechanisms. The immune response is designed to clear pathogens. So when we have an infection, anytime there's tissue damage and destruction, it could be externally or it could be within the body. That activates the immune system. People sometimes forget that the innate immune response is also responding to things going on inside the body that are injuries. But the problem comes when inflammation becomes chronic and persistent. And then at that point, inflammation can contribute to a number of diseases that are the modern ills of our society, including cardiovascular disease, diabetes and metabolic disorders, as well as cancer, and now as we move into the brain, neurodegenerative disorders, and what's going on in terms of the immune system can ultimately affect behavior, and that's what we're going to be talking about today. And in fact, this idea that inflammation in modern societies, I mean, you have to think of why do we have such aggressive immune responses? It really is about ancestral requirements, if you will, to get to reproductive age. We had to go through a boatload of pathogens to get there. And of course, there were a lot of predators around, so ancestral humans really had a very aggressive immune response. And if you didn't, you're not sitting in this room right now. In fact, this was so important to humans at the time that I'm sure human or subhuman cultures like Neanderthals and the Soviets and such to pick up their DNA when they moved out of Africa and into Europe and Asia, because the bugs and the pathogens were quite different. And so by mating with these other species, humans were able to pick up genes that ultimately allowed them to fight these infections. It gives you an idea, and they're sitting right in areas that are relevant to the immune response, and they're biased towards inflammation. So we are all kind of biased towards that inflammation by virtue of the legacy of our ancestral past, and it's now being recognized, and this was one of the insights of the decade from 2000 to 2010 in science, that inflammation is a common mechanism of disease. So it's something all of us will deal with either personally or in our research one way or another. So what is the data that suggests that there is a link between inflammation and depression? There's a very impressive literature here that is quite comprehensive, starting with the fact that if you look at patients with depression, they exhibit all the cardinal features of a chronic inflammatory response with increases in inflammatory cytokines. It's been shown in the peripheral blood and the cerebrospinal fluid. Hundreds of studies in the peripheral blood on cytokines, and interleukin-6 and tumor necrosis factor appear to be the most reliably elevated in depressed patients. You also see increases in acute phase reactants. These are proteins produced by the liver in response to inflammatory cytokines. And these molecules are designed to sort of contain the immune response and then help clear pathogens as well. And C-reactive protein, CRP, is the most reliably elevated meta-analysis of this literature. And then you have increases in chemokines, which draw cells into the site of inflammation, increases in cellular adhesion molecules, which have the cells stick once they reach the site of inflammation. And so you see all of these increases in depressed patients versus controls. Then if you go into the brain, postmortem samples from depressed patients who've committed suicide, you see evidence in brain parenchyma of activation of inflammatory signaling pathways. There is some evidence of microglial activation, that's the immune cells in the brain, and also evidence of trafficking of immune cells to the brain as well, with the presence of perivascular macrophages in postmortem tissue. Now, there's kind of an interesting wrinkle if we go back to our treatment-resistant patients. There appears to be a special relationship between treatment resistance and increased inflammation. So if you have high inflammation and you go on a treatment, a conventional antidepressant, you're less likely to respond to treatment. And as I mentioned, especially for serotonin reuptake inhibitors and serotonin norepinephrine reuptake inhibitors, there's been a number of papers on this with people with high inflammation responding poorly, and actually a meta-analysis as well. Also, if you are someone who is a treatment-resistant patient, you're more likely to have increased inflammatory markers. So this is from a paper that we published recently. Just over 100 patients participated in this study, all depressed, all unmedicated. And we asked them, during your current episode of depression, how many treatment trials have you failed? And of course, as you might imagine, the majority of patients have not been treated at all in their current episode. But of those who had been treated, if they failed 1, 2, or 3 or more trials, you see this stepwise increase in TNF, similar pattern in IL-6 and the soluble receptor for TNF. See the same thing, same trend here with C-reactive protein or CRP. The trend was significant, but there was no significant differences between the groups, although the group in the UK has recently shown that there is a difference in C-reactive protein in treatment-resistant versus treatment-responsive depressed patients. So depressed patients who are treatment-resistant have this increased inflammatory markers. The second major body of data that suggested that there is a connection between depression and inflammation is the fact that you can administer inflammatory cytokines. Typically, people use interferon alpha. It's an inflammatory cytokine used to treat infectious diseases and cancer. Also, other stimuli, endotoxin, typhoid vaccination, have been used primarily in healthy volunteers. And administration of these inflammatory stimuli causes depressive symptoms. So these individuals, after you give them inflammatory stimuli, exhibit all of the cardinal features of depressive syndrome. And from these studies, we've learned a lot about the neurotransmitters and the neurocircuits involved, which we'll spend a good bit talking about later in my talk. And finally, there's data to suggest that you can inhibit inflammation and reduce depressive symptoms. And this is primarily coming from this literature, which is looking at autoimmune and inflammatory disorders, and then giving anti-cytokine therapies to these patients and showing that the depression levels drop. Now, of course, that's complicated by the fact that these treatments are also treating the underlying disease, but take it for what it's worth. There are a number of studies they try to control for this, but it looks as if giving anti-cytokine therapies has significant effects on depressive symptoms. And then you move into this much smaller literature looking at anti-inflammatory drugs to treat depression in otherwise medically healthy depressed individuals. And that literature is a mess, largely because of the design of the trials that have been used up to this point and the multiple off-target effects of the anti-inflammatory drugs that have been used. So there's clearly the jury is out there, and we don't have enough information at this point to begin treating patients with anti-inflammatory drugs. Now, based on this massive literature, and I guess in the last 5-10 years, people got pretty excited about it and started talking about inflammation as being an inflammatory disorder. And you'd see these papers, and we were certainly part of that hype, and we had to kind of walk it back a little bit. So I want to make clear to everyone that depression is not an inflammatory disorder. And we sort of should have known this, and this is a paper, one of our early papers, that I just use this as an example. We're looking at interleukin-6 over here, and these are healthy depressed individuals and healthy controls. These are depressed cancer patients and not depressed cancer patients. And what you can immediately see is that, yeah, there may be mean differences between these groups, but this is being accounted for by a subgroup of depressed patients who are showing increased inflammation, not the majority of the depressed population. And in fact, if you run the numbers, there was a recent paper published on this, we published as well, and it looks to be that about 25 to 30% of depressed patients will have increased inflammatory markers in otherwise healthy individuals. And you start to move into populations where there are other reasons for inflammation, that percentage can increase to as high as 50%. We see this in breast cancer populations. You see that they're just represented, that there's just a little bit that goes up in terms of that percentage. So how much inflammation you have is really going to depend what particular population you're drawing from, but it's clearly a subgroup of individuals and not all depressed patients. So inflammation is only increased in a subgroup. The other thing that used to drive me crazy is the fact that these other disorders, people were talking about increased inflammation in these other disorders as well. And so increased inflammation has been shown in mood disorders, obviously, in anxiety disorders, schizophrenia, neurodegenerative disorders, medical illnesses, all in association with behavioral changes that look like depression. So this really seems to be something that's occurring across the board. And so where we stand now in terms of the state of the art is that we're looking at inflammation affecting behavior in a way that it's really not specific to any disorder. Just because we started with depression doesn't mean this is a depression thing. This is something that really is truly transdiagnostic. This is a pathophysiologic process. When inflammation is elevated, it's going to affect specific neurotransmitter systems in the brain, specific neurocircuits, and that's going to track or map on to specific symptoms. And we will walk through all of that as we go through the presentation. So before we jump into some of the mechanisms by which inflammation affects the brain, I just wanted to go over a little bit as to where this chronic inflammation comes from. And the top three are probably the most relevant to us in psychiatry, the kinds of patients we see, although the CL group sees these kinds of patients. But in obesity is associated, as many of you know, with high levels of inflammation. What happens there is fat cells grow very quickly. They outstrip their oxygen supply and they die. And anytime there's cell death or destruction, the immune system gets that gets involved. So this fat tissue is loaded with macrophages that are pouring out cytokines. We can tell very quickly when somebody comes in, and we actually sometimes recruit on the basis of BMI. The higher the BMI, the more likely the patient will be inflamed. Early life stress and adult chronic stress is also associated with increased inflammation. A large literature in that area. People now jumping into the microbiome and leaky gut and the movement of microbial products into the blood that then activate immune cells. This is a source of inflammation as well. And then, of course, chronic illnesses, chronic infections, and then we do a lot of things to patients. I happen to work in the cancer center as well, and so surgery, radiation, chemotherapy, and even some of the new immunotherapies are associated with chronic increases in inflammation. So there are many sources of where chronic inflammation can come from and ultimately influence the brain and behavior. So what are the mechanisms by which inflammation affects the brain? This is actually a very impressive literature. Comes from studies in cell culture, in laboratory animals, as well as in humans. And we could spend the entire time, and we could probably spend the entire day going over this literature, but suffice it to say that in terms of monoamine metabolism, inflammatory cytokines decrease the synthesis, release, increase the reuptake of monoamine, serving to reduce monoamine availability. Also, by effects on astrocytes, inflammation can increase the release and decrease the reuptake of glutamate in glutamatergic synapses. With the buildup of glutamate, this can spill outside the synapse and bind to extrasynaptic NMDA receptors. And that leads to inhibition of growth factors as well as neurotoxicity. And then cytokines can have direct effects on growth factors and neurogenesis and influence neuroplasticity in the brain. So there's a lot going on in terms of these neurotransmitter systems, neuroplasticity, and when you look at the neuroimaging studies and you try to make sense, and there have been a number of studies done in this area, what you see is that there are two major circuits that appear to be affected by inflammation. One includes a circuit that involves anxiety, arousal, and alarm. This circuit here is being studied mostly. We actually did the first studies in showing that the dorsal anterior cingulate was involved, but now Naomi Eisenberger at UCLA is doing a lot of work in this area in terms of anxiety, social rejection, and the sensitivity of the dorsal anterior cingulate and these other pathways. Insula, hippocampus, amygdala, all involved in this anxiety, arousal, and alarm circuit. Also, there are circuits that are involved in motivation and reward processing, which is where most of our work has been done, and that involves basal ganglia, ventromedial prefrontal cortex, and the subgenual anterior cortex, and we'll talk in detail about that pathway. If you want to step back and we try to take a little bit of an evolutionary perspective, some of us have kind of thought about why is the immune system affecting these particular circuits. And just to give us a little bit perspective from an evolutionary standpoint, you can imagine that if an animal is wounded or sick, the two things that the animal needs to do is they need to withdraw from the environment. There's a tremendous amount of energy resources that are burned up in terms of the activation of the immune system, the inflammatory response. So by targeting these subcortical structures, you end up with fatigue, anhedonia, motor slowing, and it allows energy resources to be shunted to the immune system and away from exploratory behavior. So this leads to a withdrawal response that supports wound healing and infection. The other thing a wounded or sick animal needs to do is kind of get out of the equation and sort of go on hyper alert, because if you're wounded and or sick, you could be somebody's lunch because you're not moving around, you're not as mobile, and so there is a vigilance protection against attack, and that leads to the activation of these arousal and alarm pathways. Very sensitive, these patients, when they get exposed to inflammatory stimuli. But you can imagine if this inflammation in modern times is is chronic and persistent, that these things can start to bleed into psychiatric disorders, including those involved with depression, and anxiety, irritability, and neuroticism. So just a little sense of why we think these particular brain regions, cortical and subcortical regions, are being targeted. So what can we do from a treatment aspect? What can we begin to think about in terms of therapeutic targets relative to these effects of inflammation on the brain? And probably the most obvious thing is to target inflammation. And we have done a number of studies in this area with a drug infliximab. It's a TNF-alpha blocker. It's a monoclonal antibody. We like these biologic drugs. They're very potent, have very few off-target effects because they're so specific for a single molecule, limited drug-drug interactions. And we did a 12-week randomized controlled trial to sort of begin to see if you block cytokines, can you in fact reverse depressive symptoms. This was the design of the study. 60 patients went in. We stratified based on sex as well as their level of CRP, and gave them 3 infusions, which is a standard induction protocol for people who have inflammatory bowel disease, ulcerative colitis, Crohn's disease. And then we followed them throughout a period of 12 weeks. Just as an aside, these were patients that were recruited on the basis of treatment resistance. That's the only thing that our social media campaigns were targeting. That's what we were talking to them about on the phone when we recruited them. What was amazing is the patients who walked in had very high C-reactive proteins as a group in terms of increased inflammation. They were around 50%. This CRP greater than 3 is considered by the CDC and the American Heart Association to be high inflammation. So the mean was high, and about 50% of these patients had high inflammation according to these definitions here. And so if you do the math from the number of people who are depressed in the United States, and then you look at the third who don't respond to antidepressant medication, and then you look at the half who are exhibiting increased inflammation, what you end up is somewhere in the neighborhood of 4 to 5 million people in the United States have treatment-resistant depression and increased inflammation. This is a big population of patients. This is the number of patients with rheumatoid arthritis, the number of patients with inflammatory bowel disease. So you're talking about a fairly significant problem here. So we did the study, and this was the results. And I'm surprised I didn't kill myself at this point. This was a very tough study to do because if you read the consent forms, we had to put black boxes in there. You could die if you participate in the study. These are very potent anti-inflammatory drugs. So it's a very tough recruit, and you can see there's absolutely no differences between the groups. And when we did this study, this was a study that was done when everybody had the idea that all of the patients are inflamed. Depression is an inflammatory disorder. So of course, and I was sort of talked into this by Chuck Raison, who did this study with me, which Jordan knows, and he said, well, maybe they have a little bit of inflammation that you really can't measure, and if we give them an anti-inflammatory drug, they'll get better. And no, not at all. But what we did, which I think we had to, we went back and we looked at the data and we said, well, does it matter how much inflammation you have before you receive the infliximab as to whether you respond to the treatment? And the answer is yes. The higher the CRP, the more likely you're going to respond to treatment over time. And this is sort of what it looks like. It's a little bit of a complicated figure here. We're looking at the change in the Hamilton depression score from baseline to week 12, and then we're subtracting placebo from infliximab. But suffice to say, if you're on this side of the graph, it favors placebo, and this favors infliximab, and these are the point differences on the Hamilton depression score. If you see the sample as a whole, actually placebo did better than infliximab. But as you begin to cull the sample down into those with a little bit higher inflammation, 1 milligram, 3 milligrams, and you get up to 5 milligrams, which was the sweet spot here, you see that there's a 3-point difference between infliximab versus placebo. And that's considered a clinically significant difference by the National Institute of Clinical Excellence in the UK, who kind of puts little parameters around these types of things. Also FDA in the large clinical trials. And actually for treatment resistance, this is for depression, a 3-point difference. For treatment-resistant depression, a 2-point difference on the Hamilton Depression Score is considered clinically significant. We almost hit that with a CRP greater than 3. So it really matters what level of inflammation you have as to whether you respond. And in this sort of sweet spot group of CRP greater than 5, the standardized effect size was 0.41, and that matches to a standard effect size of about 0.33 for conventional antidepressants. Just looking at the data a little bit differently here, we're looking at individuals with a C-reactive protein greater than 5 milligrams per liter, and you see this sort of almost doubling of response in the infliximab group versus the placebo group. And look what happens if you have lower levels of CRP. You actually respond much less, and in fact, you respond poorly. Some patients just didn't change at all in this group. And so it became very clear that giving anti-inflammatory drugs to patients who are not inflamed, they actually make them worse. And so this is something that really goes to these design issues that I mentioned before in previous trials. And the things you'll read in the journals are people putting a bunch of depressed patients on an anti-inflammatory agent and see if it works or not. And our data suggests it will work in some patients and it won't work in others, and it may actually do disservice to some of these patients. If you look at the symptoms that got better, so here we're starting at 12 weeks at baseline, and we're moving to 12 weeks. So if you're going in the negative direction, that means the symptom is improving. The symptoms that you see that really jump out, this is in the high CRP group, is these are the 17 items of the Hamilton Depression Score. That work and activity is anhedonia. So you see very clear separation in anhedonia. Same thing with psychomotor speed. So this is motivation and motor activity in these basal ganglia circuits, and then these changes in anxiety with that anxiety arousal and alarm circuit that I talked to you about before. And this may be relevant to some of the work that's done in PTSD here as well. So yes, you can block inflammation. I think we're not quite ready for prime time with that. We don't understand enough of what's going on immunologically to really do sophisticated, nuanced immune-targeted therapies, but we're all working on that. Where we have been particularly interested is looking at the downstream effects of inflammation on the brain and seeing if we can make some changes that are therapeutically relevant or translationally relevant based on our understanding of how inflammation affects the brain. And as I said, we're particularly interested in that circuit that's involved in reward processing and motivation and motor activity. So in order to study this, we have used patients treated with interferon alpha. For those of you not familiar with interferon alpha, it is an inflammatory cytokine itself. It's used to treat hepatitis C and malignant melanoma. And in fact, that's where we got started. I was asked to see these patients as being a psychiatrist and working in the cancer center, and it was floored by the similarity between the depressions that we saw in patients on interferon alpha compared to individuals who were just coming in because they lost a loved one or they had relationship problems or job problems or whatever. Depression really looked very similar, although what was really eerie about it is the patients had no psychological context for their depression. So it was a very kind of odd experience talking with these patients with full-blown depression. So we're using interferon alpha, and to get at this issue of reward processing, we are particularly interested in the ventral striatal region, and we used a hedonic reward test to activate that region using functional magnetic resonance imaging. The ventral striatum involves the nucleus accumbens here, this little round area, and then a tag of the caudate and putamen. That's basically ventral striatum. You move up here, you're in the dorsal striatum, and it's more related to motor activity, and these lower regions are more involved in motivation. So what we did is a gambling task, standard task. Patients get in the scanner. There's 2 cards face down, and we asked them to pick a card. They pick a red card, they won $1. Black card, they lost $1. We rigged it. Everybody won 30, 40 bucks. They were pretty happy with that. I'm getting paid to be in the scanner, so if you look to see, well, what's going on in terms of activation of this ventral striatal region, you see in the win versus lose condition, you get very nice activation in this brain region as a sample as a whole. This is a work of Lucille Capron, who was working in our group at this time. But if you go then to compare the interferon alpha treated patients versus the control, these are patients getting interferon alpha for hepatitis C, you can see the red dots are showing less ventral striatal activation compared to the control individuals. You see this on the right side, left side, and bilaterally in terms of activation of this ventral striatal region. And then if you want to then go and map these ventral striatal changes onto symptoms, onto behavior, what you see is you look at the bold activity, the win versus lose condition, the lower the level of activation of the ventral striatum, the higher the level of reduced motivation or anhedonia. You see a nice correlation in the sample as a whole, and you see a clustering of these red dots in individuals who have both low activation of the ventral striatum and high levels of anhedonia. Now, this finding of inflammation affecting the ventral striatum and reducing ventral striatal activation in association with decreased motivation has also been demonstrated by Naomi Eisenberger with endotoxin, as well as Neil Harrison in the UK with typhoid vaccination. These are acute administrations of inflammatory stimuli as opposed to our chronic administration of interferon alpha. So what you're seeing is 3 different laboratories using 3 different strategies to give inflammatory stimuli, and all of us coming up with the same results: decreased ventral striatal activation in association with depressive symptoms, including decreased motivation. Given the role of dopamine in this, in the reward processing, especially in the ventral striatum, we decided we'd go after dopamine and see if we could get a better sense of what was going on with dopamine in the ventral striatum. And again, we used interferon alpha, but this time, thanks to Jennifer Felger, we had a rhesus monkey model that we developed of cytokine-induced depression. We gave the monkeys interferon alpha, and just like the humans, they became depressed. Their depression is a kind of a huddling-like behavior, which is considered a depressive-like condition equivalent. And we put it in a probe into the brain just north of this ventral striatal region. And these are awake, alert animals that are trained to just sit there while we do the in vivo microdialysis. And these probes can both sample extracellular fluid as well as put in various drugs like potassium amphetamine that leads to dopamine release. And under resting conditions, we saw after 4 weeks of interferon alpha dramatic decreases in dopamine under resting conditions, as well as dopamine's major metabolite, homovanillic acid. And then if we went and stimulated with either potassium or amphetamine, what you see is this marked decrease in dopamine release in the interferon alpha treated animals compared to the controls. And this decreased dopamine release also mapped on to decreased motivation in these animals. And this is kind of an interesting wrinkle in the story of what does inflammation do to the brain and what does it do to reward pathways. If we just gave the monkeys a little tray that had sugar pellets in it, there was no difference between the saline and the interferon alpha treated animals in terms of how many pellets they ate. However, what it's becoming more clear is that inflammation does not affect your sensitivity to reward, so you are able to experience reward. But if you have to now, we put the pellet into a puzzle feeder, and now you have to use your fingers to get this from here to here to here, and finally down here where you get the pellet. And what you see is you see this emerging difference between the interferon alpha treated animals and the saline animals, and at 4 weeks you see a significant reduction number of pellets consumed, so that the effort for reward is reduced. And if you map that on a small sample, but this is the amount of dopamine that was produced in terms of the studies I showed you earlier, and mapping on to how many pellets were consumed. So the lower amount of dopamine you have, or dopamine release, the lower number of sucrose pellets that you will work for. So it really seems to be effort-based motivation that is effective. This work, again, I have an obsession with there being consistency in the literature, and there is a good bit of consistency in this literature. So John Salomon at the University of Connecticut has given interleukin-6 intraperitoneally to rats, and he did microdialysis, a little bit different strategy. He was putting the probe in multiple different parts of the nucleus accumbens. And within 30 and doing in vivo microdialysis, looking at dopamine and in that nucleus accumbens, which is part of that ventral striatal region I showed you before. And it didn't really matter where he put the probe in. He got consistently, after 30 minutes, this drastic reduction in dopamine in these animals after an injection of IL-6. So again, we're seeing some reproducibility, and this decrease in dopamine was associated with decreased effort-based motivation in these animals, which is John's specialty. So given what we've seen, we've become very interested in what is the mechanism, what is the final mechanism by which inflammation is influencing dopaminergic neurons to lead to this reduced dopamine. And to that end, we have just developed these midbrain organoids. These are mini brains, as some people call them, 3D human brain organoids that are derived from human-induced pluripotent stem cells. And you can grow these organoids up, and you can grow them up and have a predominance of midbrain dopaminergic neurons in the culture. We also put microglia in these cultures, or we can switch in and out. And this just gives you a picture of what these organoids look like. In green are the dopaminergic neurons, and in red are the tyrosine hydroxylase staining, which is the rate-limiting enzyme for the synthesis of dopamine. So here we're looking at HPLC, and we decided we'd treat these organoids with interleukin-6. We did that for 24 hours. And what you can see this in yellow, this is the standard just to tell us where dopamine is. Here is the control organoid, and you can barely see any interleukin-6, any dopamine being produced by the dopaminergic neurons that have been treated with interleukin-6. So we feel like we have a model system now where we can go in and understand what is the mechanism and then start to look at drugs that may reverse these changes of inflammation on dopamine. Now, based on other studies that we've done, and there is data to suggest that what inflammation does is it affects, at least in part, the synthesis of dopamine, and this is related to the effects of inflammation on tetrahydrobiopterin, or BH4, and inflammation basically reduces BH4. BH4 is a cofactor for enzyme cofactor for tyrosine hydroxylase. So if you don't have BH4, your tyrosine hydroxylase decreases, as well as your phenylalanine hydroxylase, which gives you tyrosine, which is also in the path of synthesis. So Jennifer Felger in our group said, well, why don't we just jump through all those steps and give the monkeys L-dopa, the immediate precursor to dopamine. And when she did that, by reverse microdialysis in these monkeys, she was able to completely reverse the effects of interferon alpha on stimulated dopamine release. So if you give the precursor, they're able to take it up, they're able to go the last step to make dopamine, package it, and release it. And so this can be reversed by giving a dopamine agonist or a precursor to dopamine. Now, to kind of beat this horse a little bit more, I'm going to talk about cells going to this part of the brain. So we've got a story where we see that in multiple laboratories there's decreased ventral striatal activation that's associated with reduced motivation. We know that dopamine plays an important role in reward processing in this brain region, and we show in this brain region that there's a decreased level of dopamine in several models. So how is this happening? How are the cytokines getting up into the brain and influencing dopamine metabolism? So we started to look at this. These are post-mortem samples from the brain of the interferon alpha treated animals, again treated for 4 weeks. That's sort of our paradigm. These are the control animals, not much going on. It's globus pallidus and the striatum. blood vessels and meninges. If you look at these interferon alpha treated, first you see activated microglia in the brain parenchyma, and you see these perivascular macrophages around the blood vessels, and also you see macrophages in the meninges as well. So these cells are trafficking to the brain. They're trafficking in the area of the basal ganglia and the ventral striatum, where they're uniquely poised to release cytokine that could potentially get into the brain, but there's a blood-brain barrier that can block all of that. And this kind of solved the problem. This is a paper that's a fascinating paper by Scott Rousseau and their group, and they use a chronic social defeat model. So you got a bully mouse that beats up the mouse that's participating in the study, I guess. And there's wounding involved, and there's stress involved, and this goes on for 10 days. And about half of the animals develop a depressive-like behavior, largely measured through social interactions, or they become, or they're stress resilient. They no effects on them. And then they had a control group. And what they did is they were looking in this area, the nucleus accumbens. They looked in multiple areas. The nucleus accumbens was the only place where they saw differential permeability in the blood-brain barrier in the stress-sensitive versus the stress-resilient animals. And what it looks like, you can see this in red, that's the blood-brain barrier. And what they did is they labeled IL-6 with a biotin label to the green labeled biotin. So IL-6 would show up in green. So you don't see much IL-6 around because nothing's happening to these animals, and it's certainly not getting into the brain. And then if you look in the stress-resilient animals, again, you see this very nice intact blood-brain barrier and not much IL-6 around, and IL-6 not getting in the brain. And if you look at the stressed animals, you can almost, it's very hard to make out the blood-brain barrier. So there's marked disruption in the blood-brain barrier. You see a lot of IL-6, and it's pouring into the brain. And this is occurring in the same region where we see these effects of inflammation on dopamine ventral striatal activity. So this tells us how these cytokines can get into the brain. And it's largely related to the effects of stress and potentially inflammation on claudin 5. It's a protein that's integral to blood-brain barrier integrity, and it seems to be reduced in these animals. And interesting, they did a similar study in postmortem samples from depressed suicide victims and found in the accumbens reduced claudin 5, consistent with a similar phenomenon going on in depressed individuals. So I've told you a lot about this literature where you give inflammatory cytokines or stimuli to patients or animals, and you look at what happens. And that's sort of where the field has been at. And now the field is sort of moving, and we're starting to look at patients who are depressed, which is where we started. What does this have to do with depressed patients? Does inflammation's effects on dopamine and reward circuitry, do we see this in patients with major depression? In order to study this, we've been doing resting state functional connectivity. So there are patients just sit in the scanner and they look at a cross. And we're looking at the relative activity in different areas of the brain, and those areas of the brain that are in sync, we think that they're communicating with one another. And based on this, you can begin to get hubs and circuits, and then these circuits are mapped onto behavior. And that's much of what you see in psychiatry in the journals is around these functional connectivities or task-based fMRI. We were particularly interested in this reward circuit going from the ventral striatum to the ventral medial prefrontal cortex, and that's basically the heart of the reward pathways. And you can see in this dorsal striatum, this is sort of getting up into motor cortex, and we're starting to look at something that involves both motivation and motor activity. So we were interested in this ventral striatum, and we wanted to know, we put a seed in here to do functional connectivity. Can we see changes that are in this particular circuit using fMRI and functional connectivity? And as our measure of inflammation, we use C-reactive protein. That's a marker that we feel is a very good marker for inflammation. It's immediately available. All of you can get your own CRPs, or you can get them on patients and determine immediately whether these patients are high inflammation. The levels of inflammation have already been established by the American Heart Association and CDC, as I mentioned before, and this is based on the risk for cardiovascular disease, but it seems to work pretty well for a number of disorders, including development of depression. And as I said, these are acute phase protein. It's released by the liver. And so what we wanted to know is, as your CRP goes up, your depressed patient, as your CRP goes up, what happens to the connectivity between the ventral striatum and the entire brain? So we did a whole brain study to start. And the biggest effect that we saw was the connectivity between this ventral striatal seed and ventral medial prefrontal cortex, right in that reward circuit. And so basically, as your inflammation goes up, the connectivity between these regions goes down. And this is graphed here. So the higher the level of CRP, you can see the connectivity is indicated here on the y-axis, is steadily decreasing in these individuals. And that maps on to anhedonia. We did then a mediation analysis. This was something reviewers insisted upon, so we did this. Because CRP, the point was, CRP directly relates to anhedonia. So how do you know this stuff that's going on in the brain has anything to do with it? Maybe it's just going on as a parallel process of some sort. So what we did is we did the mediation analysis, and based on this analysis, the data indicates that what's happening is you have increased CRP, you have increased inflammation. That leads to decreased connectivity in these reward circuitry, and then that leads to anhedonia. And we saw a similar story in the dorsal striatum of psychomotor activity. So motivation, motor activity being affected in these striatal regions. I was going to mention something here. This story here is well developed in psychiatry, and we know the circuits that are involved in anhedonia and depression. But what we've added here, and I think it's very important, is we've added a pathophysiologic mechanism by which these circuits can be disrupted. So when you read most of the papers, they just talk about the circuits and how they relate to behavior. They don't talk about, well, how do the circuits get disrupted in the first place? And so this is really some of the early work indicating that there are pathophysiologic mechanisms that can disrupt circuitry in the brain. My obsession with reproducibility, these are breast cancer patients now, some preliminary data. In the midst of therapy, many of these patients are depressed and fatigued and have other issues. A little bit more dorsal, the seed here, and again, this decreased connectivity to the ventromedial prefrontal cortex, and you can see the connectivity here. And as your C-reactive protein goes up in these women, the connectivity goes down. Interestingly, in this particular study, we were doing the effort task that was developed by Michael Treadway, and high effort choices, the lower the connectivity, the less likely that you're going to make a high effort choice in this particular task. So again, mapping on to behavior, these changes that we see in connectivity in a separate population of individuals. So Jennifer wanted to see if she could reverse this. And I think it starts to get to the translational relevance of some of this work. Again, we're looking now at targeted connectivity between this particular ventral striatal seed and the ventromedial prefrontal cortex. And we're doing pharmacologic fMRI, so people get scanned. And in this case, because L-DOPA had worked in the monkeys, she wanted to give L-DOPA to the humans. Makes sense. Cinemat, although we didn't put it in their brains, we gave it to them by mouth. And you can see under baseline conditions, people with high CRP, low CRP, you see this decreased connectivity in the sample here. And if you give L-DOPA, very interestingly, don't see much at all in the lower CRP group, but in the high CRP group, what you see is this dramatic return, not only to normal, but you see increased functional connectivity, suggesting that there may be some supersensitivity in these pathways related to dopamine. So this is a dopaminergic challenge. So just like within the monkeys, we're able to reverse these changes. And interestingly, this increase in connectivity was associated with a greater likelihood of making high-effort choices on the effort task, Treadway's effort task. So clearly, as we start to think about this, patients with high inflammation may preferentially respond to dopaminergic medications. And right now, when we use medications in psychiatry, we don't have anything to guide our treatment. And CRP is very easy to measure. It's a very good biomarker for inflammation. So we're very interested in point-of-care testing of C-reactive protein and then assigning people to a dopaminergic agent, of which we have a number of these, and seeing if patients respond better to those drugs than SSRIs, for example, serotonin reuptake inhibitors. This is some data to support that might actually work. This is from Maduka Trivedi's group. And they did a cut point of CRP less than 1 and greater than 1. And if you look at, this is escitalopram here, in individuals who had a CRP less than 1, 57% remission rate, which is extraordinary. You don't get those kind of remission rates with these kinds of drugs. You're usually around 30-ish, 30% more accurate. But if you look in the high inflammation group, which is CRP greater than 1, you see that this 57% drops down to 29%. This has been shown in 3 separate studies now with large community samples. This same sort of finding within this SSRI group, this escitalopram, has been used in a couple of these studies, 2 of the 3 studies. But look what happens when you add bupropion to escitalopram in this high inflammation group. You restore the responsiveness. And so based on some sort of statistics that they did, they looked at a biomarker match treatment, what the remission rate would be, at a biomarker mismatch treatment, where you get the kind of standard response rate, which is the 30% that I mentioned. So almost a doubling of response in these studies. And of course, this is post hoc, so you really have to do these studies where you start with patients that have high or low inflammation and kind of conduct the study that way. So let me sort of stop here and say that I hope I've convinced you that inflammation affects neurotransmitter systems and neurocircuits, and dopamine seems to be a major target. We didn't get a chance to talk about glutamate, but inflammation itself is a target. I think the most important thing here is that we can really begin to parse out populations of patients with high inflammation, this process of inflammation influencing the reward pathways and ultimately affecting patients' decision-making around future effort discounting and such like that. And somebody just published a paper on it showing that people who had increased inflammation, they're always thinking about the now. They're not thinking about the future. There's a kind of a shift that occurs. But we can identify these people and focus our treatments on these specific subgroups. And I think this is really, an important step towards precision medicine, using inflammatory markers is to identify patients and then to use treatments that are specific to how inflammation affects the brain. So let me just thank the people who did this work, Jennifer Felger, especially Michael Treadway and Ibrahim Haroon, and some of the earlier work, Lucille Caperon and Chuck Raison, the laboratory, and of course all the funding agencies that have helped out in this. And I want to thank all of you for your patience and attention. Thank you.
Yeah, Andy, that was just awesome. I mean, what a story. It's a vexing clinical issue, treatment refractory depression, and you bear against it the basic sciences, immunology, neural circuitry, cross-species models, translational work, interspecies reproduction, dopamine in the nucleus accumbens. I mean, this is
you have it all.
this is absolutely spectacular. I want to open it to some questions from the audience. I have one question that actually is paradoxical because it takes us away from dopamine and nucleus accumbens, which I'm very loath to do. But at one point, there was a story about serotonin metabolism and tryptophan metabolism and immune activation, shifting metabolism towards quinolinic acid, and that was a story that you didn't talk about here. I'm wondering if those models are still being examined, if you've looked at serotonin as another immune modulated target here, and how that might play into this treatment refractory.
The serotonin story is probably one of the better developed in that slide that I showed, and that's the work of Randy Blakely and their group, and they showed that especially the serotonin transporter, but it also affects the norepinephrine transporter as well. When there is inflammation, it used sort of synapsome type preparations, and then he did it in vivo. So it's been done in vitro and in vivo. So it's a very well developed story. What inflammation does is it activates pathways involving P38 MAP kinase. And these pathways, in turn, increase the expression and the function of serotonin transporters. Kind of an odd thing. And so if you go into the animals and you treat them with an endotoxin, for example, you see this very rapid clearance of serotonin from the synapse, which they've done these studies. So we, have kind of stayed away from the serotonergic, translational relevance because it is so targeted by these pathways, and you in the studies that I showed, and this has really been replicated several times, people just don't respond when they have increased inflammation. They don't respond to serotonergic agents. Now, there may be, as this field develops and we use maybe organoid models in many brains, we can begin to look to see what's going on with serotonin. Although these many brains, you have some glutamatergic neurons, you have some GABAergic neurons, around 10%, in the culture a little bit less. So you can play around a little bit with that story. And that brings me to the story with the kynureninins. So when there's inflammation, there's conversion of serotonin away from, or tryptophan away from serotonin and towards kynurenin. And we have a paper under review right now that is looking at kynurenin metabolites in the periphery and then in the brain. And what it looks like is that kynurenin is transported to the brain and then it's converted into a number of different molecules, including chynurenic acid and quinolinic acid. We're trying to tie the quinolinic acid story because it can bind glutamate receptors and cause some toxicity. So we're looking at that as a possible mechanism towards some of the glutamate findings that we have. So we're kind of going that way. Interesting, the chynurenic acid, people have always said was neuroprotective. Robbie Schwartz says, well, it's got problems. And I saw some recent data showing that chynurenic acid goes up in the nucleus accumbens in association with anhedonic behavior. And they block the quinolinic acid pathway with various enzyme mice that had the enzymes knocked out at different parts of the quinolinic pathway and left chynurenic acid alone. They were unable in all of these to reverse anhedonic behavior. They could do the forced swim, they could do some of the cognition tasks, but when it came to anhedonia, sucrose preference, the animals still continued to show these behaviors. So I think there is a lot of, there's a lot to be done in that area to really connect it to something that has relevance to treatment or has relevance to understanding how it's interacting in this kind of system that we set up. We've kind of tried to stay on a track, but that is a track, and it's glutamate. It's a glutamate story. So.
Questions?
Is there a story about ketamine and effects here? or do you think it's more downstream, ketamine and inflammation, or do you think it's more downstream on glutamate?
I think so let me answer your question just the direct. Yes, there's a relationship between increased inflammation and response to ketamine. There's also a story data showing that increased inflammation is associated with a response to ECT. And this has been shown by his name, Carlos Zarate has shown evidence of increased inflammation before treatment predicts response, and then there's Kenji Hashimoto has shown IL-6 elevated and related to response to ketamine. We think that there is a glutamate story there, and we think that ketamine is interacting with glutamate in these people with high inflammation. We didn't get into the high inflammation story with glutamate, but as your inflammation goes up, your glutamate goes up, and then that's associated with disruptions in regional homogeneity. It's just how the neurons are firing and talking to one another in close proximities and neighborhoods of neurons. So I think that ketamine is hitting a glutamate story, and I think some of the failure of these glutamatergic agents is because, again, all these trials in psychiatry, which got us into a ton of trouble, and the pharma doesn't want to talk to us anymore, except for ketamine. All these glutamate drugs are given to depressed patients without parsing out the groups. And I called up Jerry Sanacora, just published on this, with the failed, I think it was a glutamate antagonist of some sort, and basically he had no bloods to do anything on inflammation. It was a multi-million-dollar study. So it would behoove us to start to think about parsing out some of these patients with biomarkers. It's what revolutionized cancer treatments.
Biomarkers, the word of the day.
Biomarkers.
As Neil said, a really spectacular talk. So it's really nice that you're starting to understand the mechanisms, as you pointed out. So I wanted to back up one more and say what's causing the inflammation. So in the subgroup. So you talked about environmental stimuli for inflammation, things like BMI and cancer treatment and dysbiosis and those kinds of things. But I'm assuming that there's a genetic component too that increases risk. Then these people get the increased inflammation, then you get the changes in dopamine system, you get the anhedonia, et cetera, et cetera. So you have blood samples. So has anybody looked for markers of risk? I mean, one of the things that we've found, as you know, alcohol is a potent source of inflammation. That PDE4 is some variants in PDE4 are risk factors. And I'm wondering whether you have any insight into any of that.
I think that's a great question, and we have not dove into the genetics on this. I mean, clearly this is an important area. There have been, inflammatory genes that are associated with depression and other disorders related to depressive-like symptoms. We have not gotten into that, and we probably should at some point. It's always hard when you have such small samples. We do, kind of a limited number of patients, and we study them a lot. And so genetics, you need the larger populations of patients. And we tried to do this with an interferon study with one of the drug companies, where that we would get the genes, and we did find in that population, and it kind of makes sense with interferon alpha. It is a very potent inducer as well of this indolamine 2,3 dioxygenase, which converts tryptophan to kynurenine as opposed to serotonin. We found that polymorphisms in that gene were associated with the development of depression in this relatively large sample. But with the small samples that we have not gone down the road, but we're thinking about it. It's not a major strength at our institution in terms of genetics, psychiatric genetics.
Many years ago, Bob Post at the NIH and found that female rodents were particularly sensitive to dopamine agonists. You only needed half the dose to induce the same behavioral effects. And clinically, postmenopausal women are more responsive to bupropion or aripiprazole, the dopamine agonist. And so I'm wondering, in looking at subgroups, in your baseline studies of the treatment-resistant depressives, were their age and sex effects, and were they differentially responsive to treatment?
We did not see any age and sex effects. Again, small sample, hard to see anything. But with statistically looking at, is there any mediation of the response? We did not see that. But there is a recent paper that was published by Naomi Eisenberger showing that if you give endotoxin, you do see these effects in the ventral striatum. But the women seem to have a greater response in terms of behavioral response. Everything looks the same biologically, but the behavioral response is different in women compared to men. And so it's a very interesting finding, and we're kind of excited with these brain organoids because we can have male and female organoids, and maybe we can work out some of the genetics there, where we can bias the sample to certain genetic polymorphisms.
Just a little added observation. I noticed that, one mechanism for how oral contraceptives induce depression is through the kynurenine pathway.
Oh, really?
Blocks it and then prevents tryptophan.
And there are now IDO inhibitors. They failed in cancer, and there's 3 companies that have IDO inhibitors with nothing to use them for. So we're trying to create a subgroup of individuals with inflammation and increased activation of the kynurenine pathway, and see if these individuals who tend to be treatment resistant, more severe depression, anhedonia, suicidality, giving them IDO inhibitors. And the IDO inhibitors are working in some patient populations, and they've seen dramatic effects on behavior giving the IDO inhibitors to, like, kids with brain cancer, and seeing that their mood goes up despite the fact that they have, for some of them, probably fatal illness.
Great talk. I really like your emphasis on the subgrouping and trying to figure out which patients are the ones that could benefit from these inflammatory treatments or targets. In that, though, there's been mainly the studies have focused on kind of one inflammatory marker at a time, whether it be CRP or IL-6. I'm wondering if you think there would be a benefit, or if there's any research beginning on sort of a profile, an inflammatory profile, so to speak. So somebody, for example, especially something like IL-6 versus IL-10, which more of an anti-inflammatory, more of a multivariate approach to the subgrouping based on the peripheral cytokines?
I think that's the wave of the future, and to do that, you need a little bit larger sample, and the NIH has resisted us doing these kind of studies that Madhukar Trivedi and I wanted to do, so we were forced to do an R21, so it's a very small sample, and just proof of concept is what they asked. But once we get into a larger sample, then I think you can start to do microarray analyses, see if you can put together a complex sort of series of biomarkers that would then help you get a little bit closer. But again, it's kind of like the OncoDX that they use for breast cancer patients to determine whether or not they need to get chemotherapy. I mean, that's a huge breakthrough, because chemotherapy is no day at the beach. It's horrible. And so I think those are the kind of things you'd start to look for, is a pattern of gene expression that bespeaks response to particular drugs and associates with some of the stuff that I showed here today. And so we can do that. We have the microarray. We haven't done it yet, but I think we're thinking in the right direction. We use CRP, I'll just mention briefly, because IL-6 is very sensitive to what's going on before you get the sample. So if patients are stressed out and they're racing across the parking lot to get to the lab, they will have the high IL-6s. IL-6 also has a circadian release, TNF, sort of a similar thing. These are very short-lived, short half-lives of the cytokines compared to CRP, which has almost a 24-hour half-life. So if they run across the parking lot, it's really not going to have an effect on the CRP. and you can pick up the infections and other things like that with very high CRPs. But so that's why we like CRP, and we can also get it before the patients get randomized into studies. We know what their CRP is kind of going to be. We always do batch analyses of all our cytokines, but you got to feel where it is, where their CRP is.
I hand it off to Igor, and I'm going to just guess. I'm going to predict what the question is going to be about. It's going to involve endocannabinoids.
Boy, how easy it is to be profiled here.
Smoking
No,
weed all the time.
I was going to say, ditto everything everybody has said. Just a terrific talk, Andy. Thank you so much. And of course, you're right. I'm deeply disappointed that the only important signaling system, which is the endocannabinoid signaling system, didn't enter your talk, but that's for another time, right? Another series of research. But more seriously, I was really intrigued by the study you reported on the selective vascular permeability in the nucleus accumbens area, I think it was,
Yes.
in the kind of stressed, sensitive animals. Is there any data or any experiments been done on models of depressed animals and whether they have selective changes in vascular permeability? Because that would be kind of interesting if depression facilitated selective permeability.
So the Rousseau group has done a number of studies. Those animals that you saw the disrupted blood-brain barrier are animals who are depressed, so they show reduced social interactions, and actually the permeability in the blood-brain barrier as reflected by Claudin-5 expression, which is the molecule that's really integral to the integrity of the blood-brain barrier. There's a correlation between the level of social interaction, which is their measure of depression, and the expression of Claudin-5. So you get a very tight correlation. They didn't show what it looked like in the controls of the stress-resilient animals, but in the ones with the stress-sensitive, you see this direct correlation between the integrity of the blood-brain barrier, at least the molecules that play a role in that, and the level of depression. They've also shown in that model that if you block interleukin-6, you reverse the depression entirely. I should mention there was a study on IL-6, anti-IL-6, that Janssen and GSK did. We've not seen the results from that, but from what I've heard, the major effect of the drug was to reduce anhedonia. It did not hit a depression signal. It hit anhedonia. So, you start to thinking about subgroups. You want increased inflammation and increased anhedonia. And we've done some studies on those subgroups. You take people with high inflammation and those with anhedonia and those without, and you see all of these metabolic changes in lipid metabolism and glucose metabolism in this group with high inflammation and anhedonia. You don't see a thing in the other group. So all high inflammation is not created equally, which gets to your issue of subtyping inflammation. We published on some of this with glutamate, trying to take people with high inflammation and high glutamate and trying to develop those kind of subgroups that are relevant to treatment.
Thank you for talking about inflammation. This is an immunologist speaking.
Okay, great.
I'm sort of along the lines of the lady on my left. It's a beautiful data, but you silenced first violin, the orchestra keeps playing, or you silence a tuba, IL-6 or whatever other inflammatory lymphokine you take, one at a time. So the question is, did you look at the role of microbiome and brain-gut axis? Because that would be the easiest way to bring down inflammation on a systemic level. And at least on this campus, if you look at American Gut Project, you have so much of data to work on with in silico, because there are profiles of microbiomes, gut microbiomes, that are definitely pro neurological diseases or anti, and you could probably do a lot of good research in silico. Thank you.
Great question about the microbiome and all of this. Is it a source for inflammation? I've tried as hard as I can to stay away from the microbiome because that's a black hole. And there's a lot of stuff there, and I have to learn all this new knowledge and what's been done. But I think it's getting to the point where there is no choice, as you sort of mentioned. We have the patients; they're being run. It'd be very interesting to see because we're doing currently infliximab studies. The government wouldn't fund the 3 infusions, so we just give 1 infusion as a challenge. So we can look at microbiome before, after, map it on to all sorts of things related to behavior, to metabolism, to effort-based motivation. So we have all the tools from the behavioral side, from the neuroimaging side, but we just haven't, there's something about getting a stool sample that it just hits me. So I think I may have to dive in at some point, literally.
We all have to dive into that stuff at some point. Any other questions for Andy?
Hi, Scott Latender. I think we met years ago with Lucille Caperon.
Ah.
Was working. I'm an infectious disease physician, and we were working in hepatitis C and interferon at the time. But just a couple of comments about IDO. So Dr. Hong's an immunologist and other immunologists, but in the AIDS clinical trials group, the immunologists tell us that kynurenine also has immunosuppressive effects. So they actually brought it up because they were looking into these IDO inhibitors as a way of reducing the higher level of inflammation that people with HIV have. And so just making the point, I guess, that the multifactorial effects that many molecules have.
Right.
And so one comment. And the second comment is just regards to the blood-brain barrier. I was going to bring that up as well, and you nicely touched on it. But in one analysis that we did here in humans, we looked for evidence of a quadratic effect where either higher or lower blood-brain barrier permeability could be associated, at least in this case, with cognitive impairment. And we found evidence of that. And so not many people look for that, but if you overshoot, reduction in blood-brain barrier permeability, and you increase tight junction protein expression and reduce adhesion molecule expression, you'll reduce the normal surveillance of the brain that may be important for people with a neuroinflammatory process. So we can't say much. We've looked at Claudin 5 and other tight junction proteins to see if we can infer from the human data why this might be occurring. But it was interesting that having sort of low blood-brain barrier permeability was also associated with worse outcomes. So,
yeah, that's a very good point, and I appreciate that. Interestingly, there is some data, it's not yet published, not by our group, where they've looked at the movement of, I can't remember what contrast agent they use in fMRI, where they're looking to see if you get blood-brain permeability, and they see it in the incumbents and depressed patients. So there seems to be something about this permeability in this area. It's important. These things are more complicated than I certainly tell everybody. I mean, I could go into all the complications, and then that story would be a little bit different.
Well, I'd like to thank. Igor's got another question back there.
Step. Oh, I see. Okay. Exactly. Not. Right. It's an interesting concept. We don't know. We haven't gotten into it. There's no data in terms of animal models, so it's unknown. There are people that are starting to look at this with contrast agents that can get into the brain, and sort of looking in depressed patients with endogenous levels of inflammation does that map on to permeability in these brain regions. And as I suggested, there's some preliminary data that suggests that that may be the case. But yeah, that's
Well, there's obviously a lot of interest. We're running over time. Andy, I want to thank you and give you a big round of applause for your presentation this morning. I'll take one quick aside and just say for early investigators and trainees here, this is a great example. Anyone who had linked dopamine with depression 20 years ago would be eviscerated by many audiences, and ACNP is one, as I experienced it. And I think that bringing the force of biology and scientific inquiry always adds clarity to the story here, and we can now talk about it in that light. And so I'd also like to thank Andy by giving him, as our tradition here, this hat from UC San Diego Psychiatry. Wear it with pride, and next time you're out here, hopefully we won't have the monsoon-like weather, and you can actually take advantage of that. So trainees, in just a few minutes, will meet across the hall in the conference room. Thank you all for your attention this morning.